Welcome to this video in A-level biology, and here I'm looking at ATP, water and inorganic ions. Let's start with ATP. Something you may be familiar with is a nucleotide. Well, this molecule is a nucleotide and it's got three features. The first feature is a phosphate group here. The second feature is a nucleotide base, and in this case it is adenine, and it's got a lot of nitrogens. It is nitrogen rich. And then we have a 5-membered ring, this sugar, which is a pentose, and in this case this is a deoxyribose. And the deoxyribose, at this position, there will be no oxygen, only a hydrogen. And this will be in DNA. If it is a ribose sugar, then it will be part of RNA. It will only be found in RNA. And in the ribose, the position that's marked is going to have an OH, not a H. So it's going to have the oxygen, but if it's lacking the oxygen at this position, then it's a deoxyribose. Okay? And this is the basic repeating unit of single-stranded DNA and also RNA. Now, if we look here on the right-hand side, we have ATP. Okay? So ATP stands for adenosine triphosphate, and this nucleotide here is called adenosine monophosphate. So on the left here, the nucleotide only has one phosphate. On the right, ATP, it's a triphosphate, so we've got 1, 2, and 3 phosphates. And if it was ADP, adenosine diphosphate, it would only have 2 phosphates here. Now also notice ATP has a ribose sugar because this oxygen is present. So ATP does not have a deoxyribose, it has a ribose. And obviously it has the base adenine. So this is a comparison between ATP and the nucleotide adenosine monophosphate. Now, ATP is a very important energy store. So the bonds within ATP, these POP bonds, there's one here. This is called a phospho and hydride bond, and it stores a lot of energy. So ATP is the cell's energy store, and ATP contains 2 such phosphor and hydride bonds. One I've already highlighted and one here. Now, when we break the phosphor and hydride bond, energy is released. So breaking the phosphor and hydride bond actually releases energy, and the energy can be used in different processes. For example, muscle contraction, that's one use, mitosis or meiosis, DNA replication, also in protein synthesis on ribosomes, and any process which is making more complex molecules from simpler ones. For example, in A2, you may learn about gluconeogenesis, making glucose from simple compounds. So any process which requires energy. Another process might be active transport. And we're going to look at this process later on in more detail. So here, how is ATP generated? Well, ATP is generated in two ways. So on the left hand side, we have an arrow going up. So we have ADP, adenosine diphosphate, and inorganic phosphate, or PI. And when these combine, they make ATP, adenosine triphosphate. Now, the inorganic phosphate is phosphates on its own. It's not incorporated into an organic compound, a biological molecule. But when it's bonded to the diphosphate, it is ATP and it becomes organic. Now, this process here to synthesize ATP could be photosynthesis, and this is going to be the light dependent stage, and you may or may not have learnt about this, or respiration. That's the two ways we generate ATP. And you will know, if you've done respiration at A level, that respiration is split into different stages. At each stage, we are generating ATP. And so this is very important. Okay, so this is how ATP is generated. In photosynthesis, we are transferring energy from sunlight into energy in the phosphor and hydride bonds of ATP. In respiration, we are catabolizing or breaking down complex molecules like glucose. And when those molecules are broken down, the energy stored in those molecules are released and they're transferred to make ATP. And our ATP is storing that energy. So when we make ATP from ADP and PI, generally it is catabolic reactions like respiration, in which ATP is generated. And catabolic means you're breaking down complex molecules into simpler ones. And catabolism is part of metabolism or metabolic reactions in an organism. In the opposing side of this diagram, ATP is hydrolyzed, and this requires the addition of water. Okay? So when we add water, we hydrolyze one phospho and hydride bond, and we go from ATP to ADP and inorganic phosphate or PI. And this then releases energy stored in the phosphor and hydride bond for the processes that I've highlighted here. And this then is really important. As well as those processes, the energy released can be used for anabolic reactions. Those are reactions where you make more complex biological molecules from simpler precursors. Now, DNA replication, protein synthesis, synthesis of triglycerides or lipids, synthesis of starch or glycogen, all of these are anabolic reactions because we're making more complex molecules from simpler ones. And catabolism or catabolic reactions and anabolic reactions or anabolism, together they're called metabolism or metabolic reactions. So metabolism contains two branches or two different processes. One is catabolic and one is anabolic. I also like to say that when you generate ATP this way, it's condensation because water is released. When you synthesize ATP, it is when you synthesize ATP, it is a condensation because water is released. When you hydrolyze ATP, hydrolysis, water is added to break that phosphoanhydride bond. That is a hydrolysis. So water is an important molecule. It is inorganic. It is not an organic biological molecule. It doesn't have any carbons. And as you know, water, the formula is H2O. And here we've got oxygen bonded to two hydrogens through covalent bonds. And the oxygen is delta negative. The hydrogen is delta positive. So the oxygen has a slight negative charge. Both hydrogens have a slight positive charge. And this makes water polar. And what we mean by polar is that there's polarity. Oxygen has a slight negative charge, hydrogen has a slight positive charge. And so it has a slight charge. Water is an important metabolite. What that means, it participates in reactions such as hydrolysis. And when we undergo hydrolysis, we're adding water and we can use this to break glycosidic bonds between sugars, between monosaccharides. We can break peptide bonds between amino acids. We can break ester bonds between a fatty acid and glycerol. So this is hydrolysis and it's involved in breaking bonds. And condensation. When such bonds form, water is released. So water participates in biological reactions. Water has a high latent heat of vaporization. What this means, it takes a lot of energy to break hydrogen bonds between water molecules. And so to allow it to evaporate. So here I've got one water molecule and we have delta negative and delta positive. It's polar. I have another water molecule here and it's the same, delta positive, delta negative. And what I'm doing is I'm having a hydrogen bond. The hydrogen bond is a force of attraction between different molecules. And here it's between water molecules. So water molecules can attract to each other, and the delta negative oxygen forms a weak bond, a weak interaction with the delta positive hydrogen because of the opposite charges. And if you do A-level chemistry, you will need a lone pair of electrons here, because oxygen has a lone pair. In biology, we don't worry too much about that. So this bond allows water molecules to interact with each other. And this is in liquid form. In gaseous form, water molecules do not interact with each other. To turn liquid water into a gas, we need to break these hydrogen bonds. And here, because of this property, this high latent heat of vaporization, when water evaporates from sweat on the surface of the skin, it absorbs a lot of heat, cooling the body. When water in the xylem in the leaf evaporates, it absorbs a lot of heat and it then cools the leaf and cools the plant down. And this ensures that the body in humans or in animals and also in plants, there's no overheating, because overheating would denature enzymes and this would slow down or stop metabolic reactions. Water also buffers changes in temperature. What this means is that it reduces fluctuations in temperature. The reason why it does that is because water has a high specific heat capacity, and this is the energy needed to raise the temperature of 1 gram of water, in this case, by 1 degrees. So what a high specific heat capacity means, if I have a small amount of water, let's say 1 gram, the energy I need to transfer to the water to raise the temperature by 1 degrees is a lot. Okay? So in biology, the way we explain it is a simpler way because I've given you the definition of physics. In biology, we write down that water absorbs a lot of heat, but the temperature increases slightly. Also, water needs to release a lot of heat for the temperature to decrease by a small amount. So here, if we have water, let's say in a pond, and the surrounding temperature in the air increases from a cold temperature in winter to, let's say, 40 or 50 degrees in summer, the water will absorb a lot of heat energy, but the temperature of the water won't increase very much. Also, let's say we go from summer to winter, then the water will lose a lot of heat because the surroundings will be cold, but the temperature of the water won't decrease very much. So here, the temperature of the water doesn't fluctuate very much in different seasons, and this protects organisms. For example, if the temperature of the water releases 40 or 50 degrees, the enzymes in plants and animals in the water will denature and they will die. And if it decreases too much, then the metabolic reactions in those organisms will slow down so much that they won't be able to support the demands of energy in the cell, and they won't be able to produce enough ATP. And in that case, the organisms will die as well. So this is very important. Moreover, water is a good solvent. And here it's important to note that water can dissolve ions, which are charged atoms or molecules, polar substances, and the polar substances are going to be things like glucose or monosaccharides and disaccharides, DNA, which has a negative phosphate, proteins or amino acids, because the amino group and the carboxyl group in amino acids can become charged at different pHs. Let me show you how water molecules dissolve sodium. So here I have a sodium ion. The sodium ion will be surrounded by water molecules. Now, the water molecules are polar. The delta negative, the slight negative charge on the oxygen, will be attracted to the sodium. And because these water molecules are surrounding the sodium and there is weak attraction between them, between the opposite charges, this allows sodium to dissolve. Now, water being a good solvent is very important because then water can participate or water can dissolve substances that react. For example, water dissolves glucose, which is involved in respiration. It also dissolves enzymes. And these substances, when they are dissolved, can undergo metabolic reactions. Another important factor to consider is the density of water. Here is ice, and here is water, liquid. Now, water in a liquid form has more kinetic energy than in solid ice form. And so the water molecules are moving around. So the water molecules are randomly arranged. They are closer together. And also there's weaker hydrogen bonding between the water molecules. Because you reduce the temperature when we turn water into ice, when it freezes, it has much less kinetic energy. The water molecules do not move around, and they take on a regular shape, a regular arrangement. In that regular arrangement, the number of hydrogen bonds here, shown in dashed lines like this, they're maximized. There's more hydrogen bonds. In order for water molecules to form more hydrogen bonds, they need to be more spaced out. So in ice, the molecules are further apart, so ice is less dense than water. And this means that ice can float on top of liquid water. So, importance in ponds and lakes of some of the processes that we've talked about. So, for example, when we change from summer to winter and back again, you can see at the same level in the pond, let's say at the top of the pond, we're going from 8 degrees to 0 degrees. Outside, you might be going from 20 degrees to -5 degrees, a bigger change. But in the water itself, the fluctuation temperature is much smaller, and this protects organisms, as we mentioned before. Now, as you go further down in summer, as you go further down the pond, it becomes cooler. So if it's too hot at the top of the pond, then organisms can move further down. In winter, the water is more insulated at the bottom because there's more earth surrounding the water. And here you can see it's warmer at the bottom than it is at the top in the winter. And so organisms can change the depth at which they reside if the temperature is too cold at the top. It's also important to understand that in winter, if ice forms, the ice will float on top of the water, and this ice will form a protective barrier which will insulate the pond or the lake, and it will reduce heat loss. And this will prevent the water underneath from freezing. If the water freezes, then organisms will die. Imagine if the ice was more dense than water, then the ice would freeze from the bottom up, and this would mean organisms would be at risk of dying. But this does not occur. Okay, now let's look at some important inorganic ions. These are ions which do not contain carbon and they're not biological molecules. Okay, first we have hydrogen. Hydrogen is important in respiration and photosynthesis, and we call these hydrogen ions protons because they only contain a proton and nothing else, and they have a +1 charge. You may not have done photosynthesis and respiration, so if you have, you can talk about the use of hydrogen here. If you haven't, it's also worth knowing that the concentration of these ions determines the pH. So if you have a high concentration of hydrogen ions, then the pH is going to be lower. It's going to be more acidic. If the concentrations of hydrogen are less, then it's going to be less acidic. Now, if we have hydrogen ions, then it means that the pH is always going to be less than 7. So the hydrogen ion concentration will then affect the pH, and this will affect the activity of enzymes. Different enzymes have different optimum pHs. For example, protease enzymes like pepsin in the stomach work optimally, they work more efficiently under acidic conditions. However, if the pH becomes too low, then enzymes will denature. So it's important to regulate the hydrogen ion concentration. Fe2+ is an ion which is found in heme. Heme is a prosthetic group which is in hemoglobin. And at the center of the heme, we have iron 2+. So here we have hemoglobin, which has a quaternary structure. It has 2 alpha chains and 2 beta chains, 4 polypeptide chains. At the center of each of these polypeptide chains, we have iron 2+, inside heme. Now the iron 2 plus binds one oxygen molecule. So it will bind O2. Imagine that O2 has a double bond. It's going to bond to the iron, and both oxygens will bond to the iron. So each Fe2 plus will bind to one O2 molecule. So if you've got Hb, hemoglobin, and hemoglobin will have four heme groups, so it will have four iron 2 plus, one at the center of each polypeptide chain. Hb, we can bind four oxygen molecules because we've got four ions, and we will result in HbO8 because there'll be 8 atoms of oxygen or 4 molecules of oxygen. Each molecule will be bound to one Fe2 plus. Sodium and potassium. Now, sodium and potassium are very important because they are ions which are actively transported by the sodium potassium pump. And I'm going to look at the uses of this in a moment. Also, phosphate. The formula for phosphate is PO42 minus. Nitrate, NO3 minus, and hydrogen carbonate, or HCO3 minus. All these are important ions. Let's look at these more closely. Firstly, the sodium-potassium pump, which is shown here, is very important in absorption of glucose and amino acids in the small intestine. Here we have the lumen of the small intestine, and this is where nutrients released by digestion of food are flowing. So this is rich in glucose and amino acids and other substances. Now, the lining of the small intestine is one cell thick, and these are the cells. Okay, so we have one cell here, and each cell has these microvilli on the surface, these folds, because the membrane, which is facing the lumen, is thrown up into folds. Now, in these microvilli, in these plasma membrane, you find a co-transporter like this. This is the sodium glucose co-transporter. You can also have the sodium amino acid co-transporter, and these are different co-transporters. Now, it's important to note the stages in what's happening here. So number one, the sodium potassium pump hydrolyzes ATP, and this energy that is released is used to actively transport 3 sodium ions out of the epithelial cells into the blood. And at the same time, 2 potassium ions are actually actively transported into the cell. It's worth noting that anything that leaves this cell and is going to the blood, into the capillary shown here, firstly it will move into the tissue fluid, and then from the tissue fluid it will diffuse into the capillary. Now, it's important also to note that the capillary will transport anything that is transported into the capillary. It will be transported away from the small intestine because the blood is flowing. So the sodium concentration in the capillary is kept low. But the sodium concentration inside the tissue fluid, which is here, may be actually quite high. So from the tissue fluid, it's going to diffuse into the capillary, but from the cell into the tissue fluid, active transport. So this active transport ensures that the sodium concentration shown here inside this cell is kept low. And this concentration is always going to be lower than in the lumen. So what happens there is there is a diffusion gradient. And so sodium can diffuse in down this diffusion gradient into the cell. As it does this, this is coupled to transport of glucose here against the concentration gradient. So here the glucose inside the cell builds up, and because the concentration inside the cell is higher than in the lumen, The glucose needs to be actively transported. Imagine you have a waterfall. The water is moving, and this is a passive process because we are not putting any energy in to move the water. Water movement, as the water is moving, we can transfer energy from this process. So water can drive, let's say, a propeller or a turbine, and this can be used to generate electricity, or it can be used to lift heavy objects. So the same process happens here. Think about sodium. When it moves down the diffusion gradient, think about a flow of sodium. This drives a conformational change in this co-transporter, which also allows glucose molecules to move, but glucose is being actively transported. What happens here is very straightforward, because the co-transporter is what we call a carrier protein. So it's only open to one side of the membrane at any one time. Here we have sodium and glucose binding to specific binding sites on the lumen side. Once they've bound, there's a conformational change. And that conformational change is actually driven by the movement of sodium down the diffusion gradient. And so sodium and glucose are released. And now they are in the cell, in the cytoplasm of the cell. And as they move away, and as they are released by this carrier protein, which is a co-transporter, it will change conformation back again into its initial form, and it will be ready to bind more sodium, more glucose. This is how this works. Okay, so this is very important now in absorption of glucose and amino acids. The potassium here, which is being pumped into the cell, this potassium is actually maintaining the charge gradient. If we move too many sodiums out of this cell, then the outside of the cell will be more positive, the inside of the cell will be more negative, and this may represent a problem. And so to maintain the charge gradient, potassium, which also has a positive charge, is pumped in. Okay, phosphates. So phosphate is very useful in a number of molecules. So here you see phosphates are present in nucleotides. So you will find phosphate in DNA and RNA, and it has this negative charge. This negative charge allows DNA and RNA to be soluble in water. And this is very important for the reactions that DNA undergoes and also RNA. Now also we need to understand that the phospholipid bilayer has phospholipids. Each phospholipid has a hydrophilic head, shown here, and a hydrophobic tail, shown here. Now the hydrophobic tails are interacting with each other through hydrophobic interactions. On the outside surface, both here and here, the hydrophilic phosphate heads, which have negative charge, are going to interact with the delta positive hydrogen of water. And this hydrophilic interaction helps to stabilize this phospholipid bilayer, and it allows it to maintain this structure. Nitrate. So nitrate, or NO3- supplies nitrogen to the organism, and nitrate is important in synthesis of amino acids which contain nitrogen, and also nucleotide bases which also contain nitrogen. And normally this occurs in plants, so plants absorb nitrates, and the nitrogen combines with molecules formed from glucose, which is shown on the right hand side here, and this leads to formation of amino acids and also of nucleotide bases. Hydrogen carbonate. This is an important molecule, and I'll show you the process here by which hydrogen carbonate functions. So here we have a red blood cell on the left hand side, and this red blood cell is at the tissue respiring cells. So what happens here at the respiring cells is that carbon dioxide is released from respiring cells. It diffuses into the capillary and it diffuses into the red blood cell. Here it combines with water, and this combination is catalyzed by the enzyme carbonic anhydrase, and it leads to the formation of H2CO3 or carbonic carbonic acid. And I'm going to show you how this works. CO2 plus H2O. This is a reversible reaction, goes to H2CO3, and this is carbonic acid. And the enzyme which catalyzes this is carbonic anhydrase. And now that H2CO3 then can, and this is also a reversible reaction, it can dissociate proton. Okay, so it dissociates protons to make hydrogen carbonate and H+. So this is, because it's an acid, protons will be released, they will dissociate. This is our hydrogen carbonate ion. And hydrogen carbonate, HCO3- is the main way in which carbon dioxide is transported in the blood. And here it's going to be transported to the lungs. So it's very, very important to understand here that HCO3- then is going to diffuse out of the red blood cell into the capillary. To maintain the charge gradient, chloride ions are going to move into the red blood cell. So this is the process. Now imagine if carbon dioxide dissolved directly into the blood plasma, the liquid part of the blood. If that happened, it would not dissolve very, very well. And imagine you have a fizzy bottle, a Coke bottle, and you shake it and you open it. The carbon dioxide is released. Here, carbon dioxide is dissolved in the liquid at high pressure, and it can at any time it can become a gas. And if this happens inside the blood vessels, then it's going to cause a lot of blockage, and it could cause a lot of damage or death. This is why we convert the carbon dioxide into another form which is easily soluble in water, this hydrogen carbonate. Now have a look at what happens when the hydrogen carbonate, when we have the red blood cell near the alveolus. Now here, the carbon dioxide concentration in the alveolus is going to be lower than in the capillary. So what's going to happen is the carbon dioxide, the small amount of carbon dioxide that is there, is going to diffuse into the alveolus. What this is going to do is this is going to reverse all of the reactions. The hydrogen carbonate is going to combine with the H+ and it's going to form carbonic acid. The carbonic acid is then going to dissociate, and the carbon dioxide is going to, that is released here, is going to diffuse into the alveolus and be breathed out. Now, hydrogen carbonate is very important in buffering changes in pH. So it's this reaction here, because carbon dioxide can make the blood very acidic. And this is very important because it prevents the pH becoming too low or too high. And this, if this happens, it will denature enzymes, and metabolic reactions will slow down and stop inside cells. And this can be very harmful, and it could cause death. So here, if there are too many H+ ions, if the H+ ion concentration inside the blood becomes too high, that means it becomes too acidic, they will react with the HCO3- and they will be removed. So the pH will be prevented from falling too low. If there's too much OH- ions, and OH- ions, right, occur when the pH is above 7, when there's alkali, then what will happen is that they will react with the H+ here. And this will prevent the pH becoming too high or too alkali. And this is what we mean by buffering the pH of the blood. Let's go beyond the spec. So I want to talk to you about something called purinergic signaling. And this involves nucleotides such as ATP, or adenosine triphosphate. Now, some cells, including immune cells, contain what we call the P2Y2 receptor on the plasma membrane. Now, this will bind to specific nucleotides. For example, P2Y2 will be bound by ATP. Now, when this happens, this will result in intracellular signaling or intracellular chemical messages inside that cell, which will activate the cell or change its behavior. So, for example, if we have a cell that's undergoing apoptosis, which means programmed cell death. For example, in the fetus, when tissue in between the fingers needs to be removed to form the digits or the fingers, then that tissue is removed by apoptosis. Now, the apoptosis means that the cell's structure will disintegrate and the contents of the cell will be released. And this, if it happens and the contents are released into the surrounding tissue, it can cause inflammation and it can cause harm to the tissues. In order to prevent this, ATP will be released from the cells undergoing apoptosis. The ATP will activate phagocytic cells such as macrophages and dendritic cells, and it will do this by binding to the P2Y2 receptor on their surface. These cells will then follow the chemical trail of ATP from an area of low concentration to increasingly high concentration until it reaches the apoptotic cell. Then it will engulf and destroy the apoptotic cell, preventing any inflammation and damage of surrounding tissues. And so this is a very important and a relatively new area in biology. This kind of signaling is also important in certain features of regulating heart and also regulating nerve impulse. I hope you found this useful. Please also check out my other videos on biological molecules.